All righty. So I've been promising we're going to get into these biochemical pathways for better or for worse. And today is that day. We're going to start talking, or not start, we're going to talk about the first biochemical pathway that we're going to study for this class, and that is cellular respiration. Now, we'll get into this, we'll get into a lot of detail, but overview, because I think that can be helpful, is this is how we go from sugar to energy, pretty much. And we're going to talk about exactly how that happened. But this is what happened. This is the process. When I say biochemical, it literally just means exactly like it says, the biology and chemistry coming together. Remember, it's a pathway. And just like I told y'all in the last lecture, that means that it has to happen in a certain order because the products from one become the reactants for the next thing. Um, there are going to be a lot of jargon and a lot of specificity in this lecture. I'm going to try really hard to tell you what the important thing is, like the take-home message for most of these slides, because I don't want you to have to memorize. I don't expect, or I don't think it's beneficial, really, for you to try to memorize all of these enzymes and all of that. That's not really the point of the class. It's just like a little bit bigger than that. But if you have questions or if you're like, hey, girl, I have no idea what's going on, always, always, always feel free to email me, and we can work on that, and I can kind of guide you to be like, okay, yes, that's true, but that's not the point of this. So we're going to talk about cellular respiration today. So here it is. This is the big thing. Don't get bogged down in the exact chemical formulas, like what is 602 and all that. You should realize that they are balanced, which just means that there's the same number of molecules of each element on either side of the arrow. So if you look at this side of the arrow, there's 6 carbon, because here's C6. There are 12 oxygen, and there are 12 hydrogen, and there are 18 oxygen. You know there's 18 because you have right here, you have 6, and then you have 6 times 2, which is 12 over here. Then you just add those together to get that 18. And then on this side, you have the exact same thing. You have 6 carbon, because again, that 6 comes here. You have 12 plus 6, which is 18 oxygen, and then 12 hydrogen. So it's the same thing, but then you're getting energy. Now, this glucose comes from food or photosynthesis. Photosynthesis, we're going to talk about in the next lecture. So I'll give you some more information about that. But this is where, when we talk about, oh, whatever you eat, your energy gets your food. This is how this happens. So that's kind of the big thing. And this happens for every organism that is a eukaryote that has mitochondria. So all eukaryotic cells. This does not happen in prokaryotic cells because there is no mitochondria. Because remember, prokaryotic cells don't have organelles, so they don't have mitochondria, so they can't do cellular respiration. I'm going to show you an overview. Do not get overwhelmed by this. I thought this is a bit of a confusing picture, but we're going to break it down in the next few slides. So the first stage is glycolysis. And just like it sounds, glycolysis is literally, lysis is breaking. It's breaking up the glucose. So that big glucose molecule, it's splitting it in half pretty much. And so then that glucose, and that happens in the cytoplasm. And then there's 2-pyruvate, which is, again, just like half of glucose, goes into the mitochondria. All of this crazy stuff happens. And this part is really cellular respiration, or this part, like in the mitochondria. So we have glycolysis, and then we have cellular respiration in the mitochondria. And again, I realize there's a lot of things, and you're probably like, what the heck? What does this mean? Don't worry about it. I'll tell you, and we'll go through each part in the subsequent slides. But I just wanted to give you like an overview so you know where we're going. One thing that you're going to see are these NADHs and FADH up top highlighted in yellow. You might be like, what the heck is that? Do you remember in the last lecture I talked to you about coenzymes and how they help fit on the active side of those enzymes to make other substrates fit? That's what it is. NADH is a coenzyme. That's all it is. It technically stands for nicotinamide adenine dinucleotide. You don't have to know that. Absolutely do not have to know that. But I just have it there in case you're one of those people that's like, oh wow, an acronym. What does this acronym mean? That's what it means. But I would never ask you that. I don't know that. I know that DNA is deoxyribonucleic acid, but NADH, I have no idea what that means. Without like reading it. So don't fret. But it is a coenzyme. And remember, those coenzymes come from vitamins. But that's why you had to understand enzymes first, and what coenzymes were, because that's what this NADH is. And it's really important. And you'll hear me keep saying NADH. That's because that's the form it is a lot, and that's just where it has 2 extra hydrogen. And then it'll lose the hydrogen and become NAD plus, and then get it. This cycle is really, really not important. I would not fret about this. I'm just showing you, and we'll talk about it later. Like you'll see it in action. But yeah, NADH is the most usable form. You also have FAD, and FAD becomes FADH2. It's the exact same thing. It's another coenzyme. It's just a different form of a coenzyme, slightly different chemical formula. This is flavin adenine dinucleotide. But again, I don't care that you know that. But you're going to see FADH later when we talk about cellular respiration. And in case you're like, oh my gosh, what is that? I know NADH. I imagine that y'all are intelligent enough to be able to figure it out. But just to be clear, it's just another coenzyme. It gets hydrogen in the exact same way. So let's talk about those phases of cellular respiration. We're ready. I believe in you. We're going to go slow. But as always, feel free to ask questions. So first off, we have glycolysis. This happens in the cytoplasm. And so this is when you have that glucose, which remember, anything that ends in ose is a sugar. So glucose is a sugar. And what happens is this actually takes 2 ATP. Remember when we talked about exergonic and endergonic reactions 2 lectures ago, and I told you a lot of times they're coupled? This is a great example. So this takes energy. So it takes 2 ATP. And I don't know, to me, this isn't 100% correct, but to me, what I like to think about it as is ATP is like an energy unit, like a battery or something like that. So this takes 2 batteries in order to make it. This also takes FAD plus and turns it into FADH. I'm sorry, NADH. We're not going to focus too much on that part or the coenzymes. We are going to focus a lot on the ATP because that's the big thing. So you have your glucose molecule. You have to put 2 energy units into it. And what happens is by breaking it, you do release energy. You actually release 4 ATP. Because remember, that's that chemical energy that's found in those bonds. And when you break it open, it releases a bunch of energy. So the NAD plus comes in, takes some hydrogen out of the glucose, and it becomes pyruvic acid. I like to think of pyruvic acid, and for the purposes of this class, it's totally fine. Pyruvic acid is just kind of a half a glucose molecule. It does have 3 carbons. That's kind of, those dots are representing carbon, but you don't need to worry about the chemical formula and all that. So glycolysis, we are breaking glucose, the lysis of glucose. Breaking it in half. It takes 2 ATP, but you get 4 from it, and you get 2 pyruvic acids. That means your net is a 2 ATP. You do that because you have 4 ATP minus 2 ATP gives you that 2. So this is how you get 2 ATP. This happens in the cytosol or the cytoplasm. So this can happen for anything. If they have glucose, they can go through glycolysis. You do not need organelles, which does mean that prokaryotic cells can have glycolysis. If they have glucose, they can do glycolysis. That is the first stage of cellular respiration. Again, when we talk about those biochemical pathways, you have to do one stage in order to do the next stage because you need these pyruvic acids in the next stage. So, like I said, though, because this happens in the cytoplasm, this happens in all organisms, be it these little bitty bacteria, which are prokaryotic, or this big old chonkin raccoon. All of them do glycolysis. If they have cells, which all living creatures have cells, all of our cells have cytoplasm, so they can all do glycolysis. So this is one way in which all organisms can transform is a much better word, transform energy, sugar into energy. So glycolysis, it's magical, it's wonderful, we love it. And it's the first stage of cellular respiration because you have to have those pyruvic acids. Then you have this little prep reaction. It's not very exciting. It's not very flashy. It takes those pyruvic acids. They're also called pyruvate, which I realize is confusing. Who did that? I don't know. Again, sometimes these old scientists, what are they doing? Pyruvic acid equals pyruvate. I usually call it pyruvate, so if you hear me say that, that's what's going on. So this step, it's not very flashy. It's not very sexy. You're not getting any ATP here. What you're doing is you're using that NAD plus coenzyme. It's taking some hydrogen. You're using another coenzyme A, and you're getting acetyl-CoA. So literally this prep reaction just goes from pyruvate to acetyl-CoA. And again, that's not sexy. That's not very exciting. But like any biochemical pathway, you have to have acetyl-CoA for the next step. Also, notice that here we've gone from the cytoplasm into the mitochondria. So we have entered in. We are doing exciting things here. This is, we're in the organelles. So that also means that here we are now officially in eukaryotic territory. Because prokaryotic cells do not have mitochondria. So again, this isn't very exciting, but it's important, and you got to do it. So then you have your acetyl-CoA. And remember, really important, that for one glucose molecule, you actually get 2 pyruvates. Remember that? Because you go from 6 carbons to 2 3-carbon molecules, if you don't believe me. Look. Oh. You don't believe me? But we'll go back. There you go. 2 pyruvic acids. So you get 2 pyruvates, which also means you end up with 2 acetyl-CoAs. It just is only showing it once, kind of like when we were talking about mitosis. It could show it to you twice, but like it's not that it does the same thing. It would just be cluttered. But you end up with 2 acetyl-CoA per 1 molecule of glucose. And then we're going to go on to the next step. We're on step 3 now. This is the citric acid cycle. Again, because these old scientists are, crazy. It's also called the Krebs cycle. They're the same thing, the exact same thing. So if you ever hear that, or that might sound familiar from like high school or something, they're the same thing. They just have 2 names. Again, I don't know why. I assume Krebs is some person and he wants credit for it, but then they're like, it's the citric acid cycle. So what happens here is a lot of stuff that I do not care that you know. So I'm going to actually go ahead, take my little black marker, just ignore all of these names. They don't matter. you'll see all of our NADHs over here and FADH2. I don't care that you know when those come in or anything, but you should know that the coenzymes are involved here. But again, I don't care that you know the exact number. And so literally acetyl-CoA comes in, it does all this fancy stuff, and you get an ATP out of it. That's it. You also get all this NADH and FADH2. That's going to be really important for the next cycle because you need NADH and FADH2 for the next step in the cycle. But you get an ATP out of it. But remember, the cycle is going to repeat because you have 2 acetyl-CoAs for 1 glucose molecule. Because you had 2 pyruvates, and both of them went through the prep reaction and gave you 2 acetyl-CoAs. So on this step, you're getting another 2 ATP. Amazing. And remember, these ATP can then be utilized for the rest of your cell to do literally anything it wants. Pretty much most cellular reactions are in some ways endergonic, meaning they need energy. And so you need these ATP. This is how your body does its thing. They're really important. So we got 2 from glycolysis, and we now have 2 more from the citric acid or Krebs cycle. So we got 4. We're killing it. But now we're going to go to the big thing. And remember, we've made this NADH and FADH2. Really important. Because now you have the electron transport chain. This is the big spender. This is like the big deal. So we're going to go through it. Again, there's a bunch of names in here. You're going to be like, oh, NADH reductase. I don't care that you know that name. I don't care that you know this name. I don't care that you know this name. But you should know they all end in ACE, which is a good sign that it is an enzyme. Which again is why we had that enzyme lecture literally last lecture. Might have been like, girl, why are we talking about enzymes? Just like, why do you care so much? You need it. You need to understand what enzymes are, how they work, so you can get it here. Because again, it's really important. So we're in the mitochondria. And what's happening here with all of these enzymes is they are taking away the hydrogen. From the NADH and FADH2 that we literally just made. We made in glycolysis, that we made in the prep reaction, and that we made in the citric acid cycle. We've been making this stuff this whole time. And so now it's actually being used, which is amazing. You know, you hate for your hard work to go to waste. So what all of these are doing in different ways, and it's also taking in water. And literally what they're doing is they're sending hydrogen ions up here. So a hydrogen ion, if we were to draw, is just a hydrogen, like, obviously it's a hydrogen. That wasn't very fair. That was pretty dumb. That's missing 1 electron. So normally, remember, we're going all the way back here to those valence shells. Hydrogen is number 1. So normally, if you were to draw its valence shell, it would just have 1 electron in it. So hydrogen ion is actually missing this electron. So instead, it'll look, it's just a neutron and a proton in the nucleus, and there's like nothing in the electron valence shell. That doesn't really matter, but in case you're like, hydrogen ion, what the heck is that? So we're building up all these hydrogen ions. All of these enzymes are shooting as many hydrogen ions up as possible. Amazing. We love that. And that creates this kind of gradient where we have a bunch of hydrogen ions up here and pretty much none down here because they've all been shot up into this space, this intermembrane space. Notice also, because you gotta just appreciate it when you see it. What is this right here? Oh yeah, that's a phospholipid bilayer. You know it is because mitochondria have that phospholipid bilayer. We know that. We've talked about that. So all of these hydrogen ions, they're chilling up here. They're all being shot up into this until they get to this guy. Now, I told you we don't care about these other enzyme names, and we don't. They're not that important. But this one, it's important. It's called ATP synthase. And, if we break this apart, that ACE enzymes, the ATP synth means it's synthesizing ATP. So literally ATP synthase makes ATP. How, you might ask? That's a great question. I'll tell you. It utilizes this gradient. All of this buildup of hydrogen ions, they're just building up and building up and building up. Well, now ATP synthase sucks one back in, and it makes ADP into ATP by binding on another phosphate group. Now, because we've made so much NADH and FADH2 throughout these other three reactions, by doing this, we can actually make up to 32 or 34 ATP. You might be like, ma'am, excuse me, why 32 or 34? That's ridiculous. Textbooks don't agree. And actually, when we do research on this, people go into cells and figure all this stuff out. I don't know, they're really interested in it. Sounds boring to me, but to each their own. People are getting different measurements. It's not exact, but you get a bunch. Considering in glycolysis, as a reminder, in glycolysis, you got 2 ATP. In the prep reaction, you got 0 ATP. In the citric acid cycle, you got 2 ATP. So you're sitting at a solid 4, and now we're getting 32 or 34. So it's a lot, which means overall you're getting close to 40 ATP for every 1 molecule of glucose. It's pretty impressive, pretty cool. Again, 40, it's just like it's a bit of a guess, and it depends on exactly the glucose and exactly some other things that are like really, really complicated that you don't need to know. I don't really understand it all myself, to be honest. But that is cellular respiration. 4 steps. Glycolysis, we're breaking that glucose into 2 pyruvates. The prep reaction, we're going from pyruvate to acetyl-CoA. The citric acid cycle, we're going from acetyl-CoA, we're getting some NADHs throughout it, and we're making 2 ATP. And then the electron transport chain, here we are. We are making ATP by utilizing this hydrogen ion gradient. Also, just as a reminder, these 3 happen in the mitochondria. The glycolysis happens in the cytoplasm, which is why it can happen in any organism. But these other 3 happen in the mitochondria. So we're only talking about eukaryotes here. Now, when I talk about in the mitochondria, I want to go about some of this mitochondria anatomy. It's really complicated. Remember that endosymbiotic theory that we talked about, where the mitochondria we think was its own prokaryotic cell, and it got engulfed into a bigger one, and that's kind of how we think eukaryotes evolved. And mitochondria have their own DNA because they're crazy cool. They're the powerhouse of the cell, obviously. But you have your outer membrane and your inner membrane because there's 2 phospholipid bilayers here. All of these folds are called cristae. And you have all those folds because the electrons transport chain happens in the cristae, which means the more folds equals the more space for the ETC. Because one mitochondria doesn't have 1 ATP synthase. It'll have those 4 enzymes, and then a little while later, it'll have those 4 enzymes. A little while later, it'll have those 4 enzymes. And if you imagine, if instead of having cristae, your mitochondria had its outer membrane, and then it was just like, all right, here we go. And let's just say over here that your ATP, or the whole chain took up this much room. You can have a little space, and we got this much room. Okay. Have this much room. Okay, you have this much room. Okay, wait, maybe we'll put one more in there. That's fine. Right? All right, great. Now, if we can have those cristae, you got your outer membrane, and then you got all these folds happening here. Gosh, this is like call the Guggenheim, man. Like, I'm an excellent artist. If you do that, then look at how many more you can fit. You can be like, oh, all right, well, here you go. There's one. All right, here you go. There's two. All right, here you go. There's three. You can see here that we can fit a heck of a lot more. We can all agree. So that's why they have all these cristae, or these folds. The matrix is inside the cristae. And it's kind of just like another cytoplasmy area. So the prep cycle happens in the matrix. And then you know how I told you that hydrogen ion gradient, which we would represent as H plus, is in the, it's what's called the inner membrane space. And what that means is it's the space between the inner and outer membrane. So if we could, like, I don't know, zoom in really close, like right here. Here's where the prep step happens. Out here's where the hydrogen gradient is coming. The electron transport chain is out here, and then you're making ATP. I just drew so much on top of that I'm sure that was hard to follow. But this is where it's all happening within the mitochondria. Really important. That's why the mitochondria has all this. Now, I told you we were using what do you call it? Oxygen and water too. And what that means is that this is an aerobic interaction. You may have heard the difference between aerobic and anaerobic, and literally the only difference is oxygen. So aerobic means it's a reaction that needs oxygen. Anaerobic does not require oxygen. Pretty simple. So the cellular respiration is aerobic. The only way you can get all of these, 40-ish ATP is with oxygen. Now, these might be familiar too. This like verbiage might be familiar too, because this is really popular in exercise. You have aerobic versus anaerobic exercises. Are you breathing or are you not? Things like running a marathon, you can't hold your breath the whole time. You die. But for a sprint, some people argue that a sprint is an anaerobic exercise. There's a lot of sports scientists that disagree. I don't know. It's being debated. But this is something that you may have heard of if you're an athlete or with sports science. Because humans, though we breathe air, sometimes you hold your breath, or if you're swimming or something like that. Now, there is an option if you're out of breath, you don't have any breath, or some of these organisms like bacteria live at the very, 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 very bottom of the ocean where there's not a lot of oxygen. So what do? How do they get energy? They go through a process called fermentation. So we have our pyruvic acid. So that means they've already done glycolysis. And they got 2 ATP from there. And then that glycolysis gives you 2 pyruvic acids. Instead of doing that prep step to make acetyl-CoA, they then make NADH. This is also what organisms like prokaryotes do, because remember, they don't have mitochondria, so they can't do cellular respiration. They can't do the ETC because they don't have the inner membrane space of the mitochondria. So when prokaryotes do glycolysis, this is the next step. It's the only thing they can do. And here you're taking NADH and taking some hydrogen and making it into NAD. That's all it is. The result of that is 2 lactic acid. But by breaking the bond, you are making 2 ATP here. So that means that for prokaryotes, for 1 glucose molecule, you get about 4 ATP, as opposed to 40 with cellular respiration. Not near as efficient, not near as good, but it's still enough to make it work. So it's fine. I say it's not near as good, but it's enough to make it work. These organisms survive. They've survived for billions of years. So like they're fine. They're not like trying, they're not like jealous, like, oh man, I wish I had cellular respiration. No, they're like, why do you need that? I'm living my best life without it. But this lactic acid then builds up. Now, a lot of our bacteria and stuff have ways of getting rid of it. But you probably have had to deal with lactic acid buildup in your life. That's what causes muscle cramps. So when you are exercising, you obviously using a lot of your energy. You might be using a lot more oxygen than you realize, because I hope when you exercise you still breathe. But sometimes you can use up all the oxygen, especially in your muscles, because when you're exerting a lot of energy and stuff, your body is going to prioritize where does your oxygen need to go. And like your leg muscles may not be the #1 priority. So instead, like your heart muscles, it needs the oxygen so it can have enough energy, build all that ATP so it can pump and stuff like that. Your legs are great, we love them, but like it'll be okay. So that's what you get muscle cramps from, is building up this lactic acid. Now, how do you get rid of it? Have you ever like heard of ways to get rid of muscle cramps? So when I first was, doing research for this lecture, I was like, yeah, you just eat bananas or you drink some water. But that's what I've always been told, that potassium will help. Or you're dehydrated, you need to drink some water. Has anybody else heard this? Like maybe your coach has told you this or something? So then I did some research, I Google scholared it, and don't worry, we will cover that in lab if we haven't already. We'll talk about how exactly that works later in the semester when you're writing your paper. But you got to do good research. You can't just like Wikipedia stuff, because Wikipedia is very unreliable. So I looked it up. Turns out that's crap. That's not actually true. Bananas and dehydration are not the cause. They don't help with lactic acid. Like bananas are delicious if you're like, oh, I have muscle cramp, I just want to eat a banana. That's fine, but it's not going to help with the muscle cramp, which is crazy to me. I don't know where that like rumor got started, but there's been a lot of research, and as of right now, there's no scientific evidence to back that up. Just like drinking some water, it's not just because you're dehydrated. But what does help if you do have muscle cramps, what does help is a massage, and like a specific muscle massage. You might can like learn how to, like, if you're like, oh, every time I run I get like a cramp in, like, I don't know, my left quad, then you can learn exactly how to massage it, because it's not just like touching it. There's a specific way to do it. But yeah, or taking a break, which I mean, I can understand if you're like at like football practice or something and you're like, hey coach, I got a cramp, I just gotta take a break. That's what my biology teacher said. Please don't tell him that. I don't need to get emails. But yeah, that's the science behind it, which is crazy, because like I said, I just assumed that I was gonna sit here and be like, this is why the banana thing works. That's what I was really trying to find out, was like, bananas, how does that work? It doesn't, which is crazy. But this lactic acid in other organisms, it doesn't cause muscle cramps, but it does cause carbon dioxide to build up. And that is how you get bubbles in your beer. So we talked about how yeast is, when we talk about prokaryotes, how that's like the way you get all the most delicious foods like bread and beer. The reason why it bubbles is because of this lactic acid. Fermentation is going to cause it to bubble. That's also how you get the alcohol, which is, what gets you a little wasted. But like it also is what causes all the bubbles in your sourdough bread. If you look at bread, it's not just completely solid, though I have made a loaf of sourdough that was completely solid once. It wasn't very good. But it's got all the holes, and the holes are caused by this lactic acid because it's releasing carbon dioxide, which is really cool. Now, all of these things that I've been talking about, all these pictures I've been showing you, have been static images. I have a video. It's a really, really good video. It's like 8 minutes, and I cannot recommend it enough. I think it's incredible. But if I try to play it for you right now, what's going to happen is the sound is going to be all sorts of wonky because it's going to be coming out of my computer. What do you call it? Like my computer speakers, but then my microphone's going to be trying to collect it. It's not going to work. So what I'm going to do instead is you'll see, there's going to be a link for this lecture, but there's going to be another link right under it for this video, specifically the cellular respiration video. So I'm going to put that there. I really recommend you watching it because it'll be animated. So when I talk about like ATP synthase, you can actually see it moving. I think it's really helpful. So I'm going to put that on Blackboard just so you have it. The Amoeba sisters are like little, no, should say little scientists, but they're scientists and their videos are actually really good. They're super helpful. So I'm gonna put that here and on Blackboard so you can watch that. But that is the end of the lecture. I hope cellular respiration makes sense. We're going to talk about another biochemical process in the next lecture, which is photosynthesis. So make sure you get this. And if you don't, email me. Not a big deal.